So it's a great pleasure talking about the credibility revolution and ecology and evolution today. So this is David. Yeah, again, thank you for Peter for introducing me. Thank you for David for inviting me. And this is on the website. It says like ecological statistics, replications all about sort of like reproducing empirical study. It's nothing to do with statistician, but I want to convince it's a lot to do with statistics and a lot to do with empiricists like us too. Okay, from the first time I heard about the word replication or reproducibility crisis was through this paper. So it was published in 2012. In Nature, this commentary, this Glenn Begley, they and LSA replicated like a 53 landmark cancer study. It's not like, the ecological or evolutionary biology study, it's a cancer landmark study. And 90% of them failed to replicate. That's like, it's a lot, yeah? That's sort of a lot to everybody. However, Glenn, he called this like innovation opportunity, very positive take. And Semin, Glenn's actually Australian, and Semin Vizier, she's at the University of Melbourne, same place as Peter. She called it credibility revolution, so as my title. And this is because now we identified problems, which we will see some of them, now we can work on it. So we have plenty of opportunities, things, we can change a lot. And this is something we want to think together today. And I want to acknowledge my collaborator, Tim Parker, Whitman College, US, and Fiona Fiedler, University of Melbourne. Fiona is a philosopher of science, Tim is behavioral ecologist like myself. We've been working on this reproducibility, open science stuff last 6 years together. Okay, I'd like to challenge ecologists, ecostatisticians today, and then my talk will be 3 parts. First, we want to think beyond computational reproducibility. Second, we want to define what's replication statistically. And third, once we define it, then we want to think about what can we do? What should we do? And I'll be very provocative, so I'll make some sweeping conclusion and something say something provocative or extreme. Let's see. So beyond the computational reproducibility. So it's, ecologists are ahead of the game. So we achieved data archiving in 10 years ago overnight because 5 powerful journals in ecology and evolution, they decided that archiving should be compulsory. And 10 years later, now, I don't think you cannot publish without archiving data in ecology, which is like a really fabulous change. That's not the case for the psychology. That's definitely not the case for medicine. Medicine is a bit difficult because some data were confidential, anonymity issue, and minor. But the ecology, this is fantastic, yeah, in evolution. But we know this is not enough. Yeah, what's next? Next obvious step is, yes, we need to do the compulsory code archiving. And the code archiving not only includes analysis code, but the data cleaning code. This is where I think it's lots of mistake and confusion happens as well. And I'm quite surprised that's not compulsory yet. I quite often have to ask during a review process, can I see the code, So we need to think about how can we make all the journals, should be all compulsory to provide code. Okay, let's assume we achieve this. Now we have all the data and the code, so we can computationally produce, but we need to think further. What's further ahead is, once you have a data and analytic code, you can actually think reanalyzing and doing it differently. And psychology, they had this project, many analysts project, and 29 different teams analyze same data set. You might have heard of it. So this data was, darker skinned footballers get more red card, and they came with those different groups came up with different conclusions with the same data. And really cool thing is we are now conducting many analyst ecology and evolutionary biology version, led by Hannah and the team. So Peter's there, Fiona, all those people involved in this is conducting this. It's really excitingly about 70 different analysis teams involved. So watch this space and we'll find out what we will find out. Okay, movement I want to advocate is, so we should be able to update our analysis and the conclusions, and this should be citable. It's very difficult at the moment to publish or amend those things, and we should be able to correct our mistakes without punishment. At the moment, if I make big mistake, I have to retract paper. I wonder, like, that's not an incentive at all, because sometimes mistake may be small. We want to update, augment data. So we need to think about the mechanism. We can do this much easier. And other science, science is self-correcting. We need to promote, incentivize this self-correcting mechanism. And others should be reanalyzed, reinterpret, and all those should be all citable, publishable. But at the moment, I found this Twitter post, Frederick Utfeld, his paper, we'll see it later. And he asked to the tweeters, have you published the corresponding article criticizing another paper? Many of people are saying actually, like half of them are afraid, doing this. But it's like a sad state of affair, because science, we should be able to criticize freely to improve our knowledge. Yeah, so we need to do something about our state. So one of the suggestions I make, you might have a lot of suggestions, some business idea, that journal called Nature. What I didn't know was like there's a 54 journals in Nature at the moment, including Nature, Ecology, and Evolution. So I'm gonna talk to Nature, Macmillan publication, now Nature Springer. Can we publish Nature corrections and updates, or Nature analysis and interpretation? When this goes well, maybe they can publish Nature collections and updates in Ecology and Evolution and all other journals. And then Nature can double their Nature journal family. Anyway, that's an idea. So let's move on to the main topic of this talk. What is replication? You think replication is easy. There's a study, you replicate and see, you do the same study again and see whether results are same, nothing difficult about it. But I argue it's actually statistically defined replications are a lot difficult than than you think. Actually, till last year, there has been no formal, certainly there are no consensus on statistical definition of replication, surprisingly. Have you thought about this? That's really surprising. And this part of the talk, I'm going to rely on, there's a wonderful paper came out last year in Psychology, so you might not read it, but you probably surely heard of Hedges, and his student, Jacob Schauer. So he actually, in these papers, they define what is replication statistically for the first time properly. Okay, so the acknowledgement, and if you wanna, he has a wonderful talk on the YouTube, so you can click on to see it if you want to know more about this issue. So this was a keystone or a milestone paper in a replication crisis, because Open Science Collaboration led by the Brian Nosek, the director of Center of Open Science, they replicated 100, good famous psychological science studies, and 100 of them. Yeah, you've probably seen this, and only 39% replicated. That's quite shocking. So it's, let's pay a little bit of attention. This is original effect size and replicated effect size. If they replicate, they should be on this one-to-one line. That's only about 30% of them there. Lots of them are smaller effect size. Yeah, on average, replicate study had half of the effect size. That's sort of alarming. Those 100 good studies in psychological science do not replicate. And on the same year, 2015, Tim and I got together, this is wonderful what psychologists doing. Can we do the same in ecology and evolution? We should be doing this 100 famous study in ecology, 100 famous study in evolutionary biology can we replicate? I mean, it's a lot to ask, but we thought this was a great idea. But once I'm reading Hedge's statistical definition on replication, I think I was a bit wrong. So this one thing to notice is this is like 100 replication, 1-to-1 replication. Actually, it turns out this is not so good design. So I was wrong, but I haven't told Tim about it yet. I will tell Tim maybe tomorrow, but I tell you today. Okay, so final product perspective replication. This gives you sort of like a flavor or intuition of what replication is like. So this is final plot. This is like a mean meta-analytic effect size. Each of the data points, the different empirical independent studies. Precision, higher sample size, high precision, lower and lower sample size. And appropriately effect size HSD, standardized mean difference. Let's imagine the paper we needed to replicate in Nature was this one. It has a very large effect size, 1.5. And to replicate, we made it high power. So this was a replicated study. Okay, actually in this psychology, Brian Nosek's science paper, their power was 95%. You know what happens when you have a high power, you actually get a small effect size because it's converged to true estimate, yeah? And actually the smaller original ones far, far away. So if you try to replicate the original effect, you're probably not gonna do it. So it's very important to think about true effects and estimated effect. We want to replicate the estimated effect or true effects? I would say we want to replicate true effects, yeah? So this is a little bit of introduction. Now I'm gonna formally define what replication is. So replication delta for the HSD, or it can be just anything. So Hedges in original paper, they call it theta, some parameter of effect size. The original minus delta replication. So, delta is the true effect of treatment. So if you have lots of sample size, you can estimate this. And V is a sampling or estimation error variance. Okay. And, replication, if it's exactly replicate, lambda should be 0 because those should be 0. Delta original, delta replication, both effects should be 0. And you can't really delta, you can't really get it, but we can get the estimate is D. So D original minus D replication. So Q statistics actually, it's known as in the meta-analytic literature, Cochrane's Q test. And the FEN2 study case, chi-square, Q follows chi-square distribution with degrees of freedom of 1. So you can do the statistical, significant test. We can reject, homogeneity, like replication, not replicate, and so on. However, we argue, or Hedge argues, exact replications unrealistic. Why is this? Yeah, it seems like a good idea. However, exact replication, even mathematical physics people can do it. Tim and I talk about, physics, they don't have a replication problem, but actually they do. Here's a forest plot of the estimated Planck constant. This is something, Max Planck, something to do with the proton energy in quantum physics. And if you see it, those are different labs, different estimate, 95% confidence interval. Sometimes what you can see is confidence interval do not overlap. So actually, even mathematical physics, they cannot get lambda equals 0. So they can't do exact replication. And this is the same for this paper, speed of light, universal constant, Avogadro's number, all those constant, it's constant, they can't even do the exact replication. So we argue, you can't really do the exact replication. Replication should be approximate replication. So lambda should be lambda naught, which is a negligible difference. How, definitions are the same, but it shouldn't be 0 because even, mathematical physics people can do it. Actually, mathematical physics people came up, negligible difference should be lambda naught, which is 0.25. And if you have done the meta-analysis, you recognize I square of 20%, meaning all those observations, we allow 20% difference between all the variation, 20% coming from difference in the between studies. 80% can be attributed to error. So that's a negligible difference in physics. And actually, Cochrane handbook, meta-analysis in medicine, Cochrane handbook says I-square 40% can be negligible. That's all randomized control trial that translate into lambda naught of 0.66. So what about then ecology then? Do we have lambda naught? We know, but I suspect we don't have any consensus, and Hedge argues those you have to come up with, field has to empirically come up to a certain agreement. Okay, we don't have one, but I suspect it will be probably larger than medicine one. Yeah, so that's something like we can discuss. How we can come up with this lambda naught for ecology. It's probably different within species, across ecosystems. So lots to think about. But approximate replication, like Hedge suggests, there is a rule of thumb for this, because so this is the original delta. What would be the negligible difference? He argues biggest negligible difference must be when delta replications around very close to zero, because if you smaller than zero, the signs are different, and you can't really say if it's signs difference, it can't be negligible. Yeah. So biggest negligible difference should be around zero when the lambda replication. And it turns out this is the most powerful assumption for rejecting non-replication or accepting replication if you formulate all those things in a null hypothesis testing framework. And this is something to lambda naught, you've seen this several times. So delta replication replaced by zero. And this value determines statistical power of replication. But I want to go back to the Brian Nosek's paper. Now, with this definition, if it's the signs the same, we say we replicate it, suddenly it's not looking that bad, because 75% of replication, if signs the same, those 175 of them, we could say replicated. Okay, let's go back to this formula. If you look at the formula, this determines the replication power, okay? And the replication of the power of original study is determined by this. And if you compare this is always smaller number, yeah? Or power will be smaller because of this term. This is the extra term. So what does this mean? If you want to achieve, if you have original study has 80% of power, in such case with, for example, Hedges D of 0.2, that's a small effect, but most of ecological study have a small effect, evolutionary biology too. This is a respectable effect with 349 sample size. And if you have replication study has the same sample size, you're only gonna get 51% of power. To get 80% of power for replication, you actually need each of them to have n equal 807. That's a lot of sample size. We are pushing. I study house sparrows, and you can get about 200, but not 807. Most surprising conclusion from this formula, though, is power. If you want to get the same power in replication as original, you actually need the infinite number of sample size, because if you see it, only when you have an infinite, this V becomes 0, and those two formula become the same. So conclusion is N dictates max power, replication power in N one-to-one comparison. So if you go back to with this definition, I don't think Brian could have said none of this didn't, rejected non-replication. So we can say it's probably 100% replication. In such case, there's a no replication crisis in psychology. I solved Brian's lifetime issue. So maybe that's that. But that's, that's sort of worrying issue continue. But how we can approach this replication crisis, not one-to-one comparison. So that's the last section. I have 5 minutes to go. I'm gonna go very quickly. So what should we do? Some paper idea, I talked about, power of the study is always unreliable. How do I say, most of paper, estimate of current effect size is too large. In such case, power analysis show we need less study, less sample size than we need. So we will like waste time and effort. So I'm gonna provocatively suggest we should ban power analysis forever. And we could write paper called A Farewell to Power, which is very similar to the paper I wrote several years ago, A Farewell to Bonferroni. Another idea coming from is do not do one-to-one replication study unless you can go really big. This Nature paper, including Frederick Gutfeld, came up, and this study was fantastic replication efforts. 900 fishes are involved. They basically said the changing acidification in the sea does not impair the behavior because the original paper said it does. But under this Hedges definition, I don't think they could have like rejected the, replication, didn't replicate, yeah. So, but something you can do this, but it's enormous effort. Instead, I think this is more, I think what we ecologists and evolutionary biologists should be aiming for is a globally distributed experiments. And the most famous one is NatNet. And they have like 100 different locations. They change the nutrients of soil, what happens to the plant. And their aim is not replication. Their aim is quantifying heterogeneity across the, because we are interested in this heterogeneity. This heterogeneity, by the way, it translates to lambda naught. So actually, Tim and I was very, very excited. We wanted to quantify lambda naught for at least in this context. However, they don't really make data available, so that's something we can change, because it's really important to get lambda naught for ecology. So another idea, maybe not everybody can do, it takes lots of effort and coordination. What's happening usually in the ecological evolutionary topic is usually people working on the same sort of topic, different ecosystem, different species. Currently, those empiricists and those meta-analysis, they synthesize, and some of the study may never publish, but the open synthesis community, we suggested maybe what you can do is we can make the community online somehow. We don't know. I want to get some opinions, ideas from you. You make the community as a whole, all the people working on the same topic get together. We do synthesis together. Some of them get published as primary research, but some of underpowered study might not, but they are part of evidence synthesis and they get all the credit. So how we can like encourage this kind of thing. So quickly, 2 more minutes left, 1 more minute left actually. So the Brian Nosek repeated again the replication one, 21 study in Nature and Science paper in the psychological sciences. This time they changed the replication criteria, the same sign, that's 67% replicated. You might remember my earlier slide. What most amazing from this study was actually before replication, they asked experts, would it replicate? They did survey, some prediction market, they have to bet the money to see which one replicate. And it seems like they almost perfectly, perfectly predicted which one replicated. Belief, this is 100% replicate, 0%. So experts know what they're talking about. And actually, the Fiona and Hannah, next speaker, they are part of this replicates. Replicates is they're asking experts whether this sort of like a paper would replicate, but in the social science literature, they haven't done the ecological literature. Can we do the similar sort of thing? Yeah, 1 minute to go. We can ask, so they're asking the psychology paper, economics paper, would it replicate, cross replication? But in an ecological evolutionary biology paper, we can ask a couple of different questions to experts. Would it replicate across time and space, conceptual replication? Would it replicate in a different species of expert, what we call quasi-replication? So we can a lot to do. We can do this kind of survey to map out confidence of our science across ecological studies. With that, I want to finish my talk. How can we facilitate all those things I talked about in section 1, and how we can conduct all those ideas I just talked about, and most importantly, how we can make ecology and evolution most robust and progress science among all the science. Thank you very much for your attention.